changing it to RNA, and we were like T equals A, but sometimes it equals U.
Oh gosh.
C and
Yep.
G is the ones that flip flop when it's going from DNA to RNA.
Yep. So okay, so DNA. You start with DNA in your nucleus, like ball up your fist or whatever. That's your DNA in your nucleus. So the DNA has to stay in the nucleus because it's like the master copy. So what comprises the master copy is A, T, C's, and G's. So then when you go to try to make a protein, that's outside the nucleus. So you got to keep the DNA in the nucleus so you make a messenger, and that's RNA. When you go to RNA, you replace the T.
Okay, hold on. So DNA is the master copy.
Yep.
And outside cell need to make a messenger.
Yep, exactly. So, okay, let's see if I can open this. Okay, so the messenger is called RNA. And you replace the thymine with a uracil. So instead of a T, you have a U. Yep. So, what's this going on? Okay, so I should be able to draw. Okay. So inside your DNA. Okay, so then you go to a protein, and that messenger RNA goes to make the protein. The proteins now have a different code, which is why it's called translation. So you transcribe DNA to RNA, and you translate RNA to protein.
Okay, so transcribe DNA to mRNA, and the proteins in mRNA make tRNA? Is that what it is?
The mRNA is the messenger, and it goes to the ribosome to make the protein. So the protein and the RNA are two separate molecules. Okay, I'm making you a little video so you can watch this back, but you have your DNA that's in your nucleus. It sends out a message that's like your other hand coming out. That's the message. And then make another fist with your other hand. That message then hits the ribosome, and then it produces a different message, which is the protein.
Okay, so we transcribe the DNA to make the messenger RNA, and then we translate what?
Translate the RNA to protein.
Yeah, that was covered in the other biology class that I only go to every other day.
Yeah.
Too bad both classes aren't doing the same thing. I would maybe learn it, right?
Right. Yeah, that would be helpful. I'm trying to figure out how I made this document for you. I would draw in there, but I can't figure out how I made the document.
No.
It's like the program's like free 7-day trial now.
Okay, so what are the nucleotides?
Okay, so the nucleotides is your DNA. So you have so it builds the rungs of the ladder. So if you imagine DNA is a double helix, which means it rotates around itself, but essentially it's your two hands next to each other being connected by the bases in the middle. And so if your fingers were to reach out, if you bend both of your pointer fingers, that becomes your bases in the middle. And
so the nucleotides are the rungs?
Yep, exactly.
And the bases are the nitrogen bases?
Yeah, so okay, so a nucleotide is the rung along with its side portion too, the backbone, is the whole nucleotide. But each nucleotide is different depending on what base it has on it, and there's 4 different types of bases. So there's technically 4 different nucleotides.
Okay. I think I got that.
Okay. So we take our bases, that's why we're zooming in just on the base, because the backbone is the same in every single nucleotide. So we just look at the bases, and that goes in the middle of the DNA.
Okay.
All right, so in the 1920s, it was believed that these nitrogen bases occurred in all living things in the same repeated pattern, such as ATC, blah, 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 blah. If this were true, then DNA would not be the hereditary molecule because there's no diversity. So you can't say that a pig is a pig and a cow is a cow and then have them have the same molecule if you're like, that you're trying to figure out what made the pig a pig and what made the cow a cow.
Okay, so it can't be the same.
So it can't be the same. Yep.
In order to make us different.
Exactly. Yep. So with the same repeated pattern in all species, DNA would not provide the variety needed for the molecule containing the genetic code. After World War II, the biochemist Erwin Chagraf made some major discoveries about the nitrogen base in DNA. His research revealed the percentages of each A, C, T, and G found in an organism's DNA. The table below includes some of his data. All right, so now this is what you had discovered, Mom. You saw that A's were the same as T's, pretty much down the board, like equal to each other, but not the same across species.
Correct.
Okay, and that's the question number 1. What observations can you make about the data table? And that's what I wrote here. The percentage of A is similar to the percentage of T. The percentage of G is similar to the percentage of C for all organisms. And then I guess you could add in, like, but not the same across organisms.
Okay.
Okay. What does the data show about the makeup of DNA for different species? Explain. So I just said here the makeup of DNA depends on the species. So the makeup, meaning A, T, C, and G, is like your mascara and your... I think the word makeup is funny, but yeah, so that's different for each organism.
Correct. All those numbers are different even though they're related.
Right. Even so, A is related to T, C is related to G, but A for yeast is not the same as A for humans. Okay, very good. Hey, little one, stop. After seeing data like this in the 1940s, what do you think researchers concluded about the DNA's potential to carry the genetic code? So they're saying, like, remember what we were saying about the DNA needs to be different in order for it to be the genetic code?
Uh-huh.
So we saw that. That's confirmed. Since each organism is different and their genetic makeup is different depending on the organism, DNA could be the genetic code that determines what an organism looks like.
Okay, so they're coming to a conclusion here that the genetic code is in the DNA because it's different yet similar.
Exactly. Yep. Very good. Yeah, and it's kind of the logic behind this is sort of like, since A equals B, then B must be C. Since the DNA is different and organisms are different, DNA could be the thing that makes organisms different.
Okay, but at this point it's still a guess, but a very logical guess.
Yeah, right. Yeah, there's a lot more studies since the 40s that have
Proven it.
right.
So they kind of made a hypothesis and are kind of doing experiments after that to prove this hypothesis, and it's being proven.
Yeah. Yeah, and that's the thing about science too, is like, here's one piece of data and you're like, cool. Then to make sure this is really the case, let's make sure that if we were to, I think the other experiment they did was to look at where shoot, I have to review, but it's something like looking at where nitrogen ended up in organisms to see like if it was passed down, if the same amount of nitrogen ended up from mother to baby, and it was similar for DNA, but it wasn't similar for proteins or carbohydrates. And they like can like radioactively tag nitrogen to see where it ends up. Okay, but that's kind of like, that's what my 9th graders, that's the experiment they looked at to figure out that it was DNA and not proteins or carbohydrates. All right, so before concluding that the pattern seen in the data is universal, which other species would you want to test? Why? And I've been talking about chinchillas, and then I also think dogs would be interesting to see like different traits and colors if they have different genetics then.
So this one here is just him picking something and telling why he thinks why it's interesting to him.
Yeah, maybe he's like, whoa, birds are different. I wonder if a ostrich is different than an emu. Or he could say any animal, basically. That's probably the easiest thing for him to answer.
Okay. Sounds good.
Okay. So this next little bit is a little bit of math, but what you're doing is you're looking at the ratio of purines to pyrimidines. And so A and T, or A and G, are both purines. And T and C are both pyrimidines.
So what is a purine and a pyrimidine?
So if you go back to the top, you can see that a purine has 2 rings and a pyrimidine has 1 ring. And imagine you're lining up a ladder or you're making a bracelet or something, and along the bracelet it's 3 wide. So you want to make it consistent 3 wide. So basically the 2 rings have to line up with the 1 ring, and then vice versa, from the left to the right. The 2 rings line up with the 1 ring. So you got purines and pyrimidines are always base pairing together.
Oh, so that's why A is always with T and G is always with C.
Very good. Awesome. Yeah, exactly.
Okay.
Nice.
Because we have to be in 3s.
Yep, exactly. I mean, and in 3s, but if you want to think about it, like the distance across from one side of the bridge to the other side of the bridge has to be the same if you're going to line those 2 tracks up. If it's going to be like a railroad track and you line them up, it has to be the same across. So but what you can see from the data, what they want you to see is that if you were to add A, let's just look at the Mycobacterium tuberculosis, the first row. A plus G, 15.1 plus 34.9 is going to equal 50. And 14.6 for the T plus 35.4 for the C is going to equal 50.
So all the A's and G's equal 50 down the line?
Down the line.
All the T's and C's equal 50?
Exactly.
Without doing the math?
Yeah. And they, in this data table, they only want you to do, for this worksheet, she wants at least 4 species.
What do you mean? Oh, at least 4 species.
So she wants you to...
calculate the ratio.
Yeah.
Okay, so we have to pick 4 of them.
Right? Yep, totally. So then what can you conclude about the purine pyrimidine makeup of DNA? It's about 50-50.
Okay. All right, so she just wants us to do math, not a algebra quotation like you got here, but the actual math, and then tell us that it's a 50-50 ratio.
Right, exactly.
Okay, so that's what she's telling us.
Your data table is going to end up being like, if you say, list all your 4 organisms down on the left, and then at the top you're going to have 1 column for A plus G, and you're going to have 1 column for T plus C, and it's going to be 50-50-50-50 the whole time.
Okay. Got that.
Cool. Awesome. So then piecing it together, the conclusion here is that Chargaff's data was a central piece of evidence used by James Watson and Francis Crick in 1953 to successfully describe the structure of DNA. So actually, you know what's really interesting story about this all? James Watson and Francis Crick, they ultimately like synthesized the final layout of DNA, everything down to like how far apart the rungs of the ladder are, to how much the curvature of the DNA goes, to where the A's and T's are on the inside and the sugar phosphates on the outside. And I guess the way this is written, like, they're using Chargaff's data, but they also use Rosalind Franklin's data, which is just sensitive, because here's a woman working in science in a lab, putting a lot of blood, sweat, and tears into her work, and then having it be used without permission from two of her colleagues, without including her in the discussion. It's just pretty...
So that's why you wrote that up there?
Yeah. I think it's, like, honestly pretty lame that whoever wrote this worksheet could talk about this story and in this day and age not mention Rosalind Franklin, because it's like, well, kind of a big deal.
She might have in class. Like I said, I'm coming in on this is a piece of work that he has not done, and when I meet with him on Tuesday, I have to get him to finish it.
Oh, I see.
And I have not sat in class. I don't know what she covered, what she didn't. This is old work.
I
so
see.
She's way past this work already, so she might have covered that in class.
Okay.
And knowing Miss Jerwich, she probably did cover it in class.
Okay.
Yeah, because she's very she's very hippie-ish.
Okay, so she would have known that. Well, so
yeah, I think she would have known that.
Okay, that's cool. All right, I got about like 7 more minutes, so let's before I gotta be on my way.
Okay, we only have one question left.
Okay, here we go. So refer to the diagram on the right. What do you notice about the arrangement of nitrogen bases? So I guess you could also say they're on the inside. So but then you could just notice A pairs with T, C pairs with G. Purines pair with pyrimidines. That's probably your 3 bits there.
Is that an equal sign between the T and
yeah.
the A up there?
Yeah, and it's basically representing bonds. So if you go back up to the top, it's kind of neat. I mean, it's like this oxygen, I'm going to give you way more information than you need to know, but the oxygen is kind of negative and the hydrogen's kind of positive, and so they like to hold hands. It's like opposites attract, right? And the nitrogen's kind of positive or negative and the hydrogen's kind of positive, so they like to hold hands. So that forms like a hydrogen bond.
Oh, is that what we're looking at, is the hydrogen bond?
Yep. And so do you know that? Does that ring a bell for you? Hydrogen bonds?
No.
Oh, okay. Well, it's kind of like there's stronger bonds than other bonds. It's cool that these are sort of weak because it means that you can unravel DNA can unzip, basically. The left side can come apart from the right side, which is why you can replicate DNA in order to have a baby or to make a new cell. You know, like if you're going to make a new cell.
So that's what meiosis and mitosis is in my other biology class?
Yeah, exactly. And
because we're ripping this apart?
Yep, exactly. And both processes rip the DNA apart. They unzip it. And you even need to unzip it in order to make RNA, too. So it's pretty important that it can be unzipped. It's like you wouldn't design a pair of pants without a zipper or a jacket without a zipper. Well, you could, but it would get complicated. And then the other thing to note is that, mitosis in my toes, meiosis in my babies. Meiosis in me babies. Mitosis in my toes. There you go. You'll never forget it. If you get a scrape on your toe, your cells will make more cells by mitosis. If you need to make sperm and egg, then meiosis will make gametes or babies. Half babies.
Okay, so meiosis is the sex one.
Yeah.
And mitosis is the body one.
Yeah, exactly.
Okay.
Yep. And then the last little bit here, how do you think Chargaff's data helped Watson and Crick to predict that DNA looks like this? And I said it helped to show what the inside of the ladder looked like in terms of bases, like which bases paired together.
Okay. Sounds good.
All right.
Thank you for all your info on that.
Oh, you're welcome. Yeah, you're welcome. I made a little video. It's real slick what I can do, and I'll send it to you.
Okay.
So if you need to review it, It's all in there.